Introduction. Yes, glaucoma often runs in families, but the story is far more complex than a single glaucoma gene. Having a first-degree relative, parent, sibling, or child with glaucoma raises your own risk dramatically, by roughly 4 to 9 times compared with the general population. In other words, family history is a very strong warning flag. However, most cases of glaucoma are not caused by one single inherited mutation. Instead, glaucoma is usually a polygenic, multifactorial disease, meaning that dozens or even hundreds of common genetic variants each add a little to risk. And environmental factors, age, blood pressure, steroid use, etc., also play key roles. In this article, we unpack the genetics, identifying the handful of rare genes that can cause glaucoma on their own, and explaining the vast network of other genes that subtly raise risk. We also explore how genetic risk varies among ethnic groups, what exciting new genetic tests and treatments are on the horizon, and what patients should do today with family history or genetic test results in hand. Monogenic glaucoma, when one gene drives the disease. A few glaucoma genes follow classic Mendelian inheritance, like sickle cell or cystic fibrosis, especially in early onset cases. These are relatively rare, but have very high impact. We highlight the major ones: MYOC, myocilin. This was the first glaucoma gene discovered. Mutations in MYOC cause juvenile and adult primary open-angle glaucoma, OPTN. In juvenile-onset glaucoma, ages 3 to 40, MYOC mutations appear in roughly 10% of patients, up to 30 to 36% in some early studies. In adult POAG, onset after age 40, MYOC mutations account for about 3 to 5% of cases. These mutations act in a dominant way. If you have one bad copy of MYOC, you have high lifetime risk of glaucoma. For example, a common MYOC mutation called TLN368-TIR is found almost exclusively in people of European descent and by itself gives a very high risk. Population studies show that carriers of this variant have about a 7-fold higher odds of POAG than non-carriers. Not everyone with the mutation gets glaucoma, illustrating that other factors matter too. OPTN, optineurin, and TBK1, tank-binding kinase 1. These two genes are linked to normal tension glaucoma, NTG, a form of open-angle glaucoma that occurs even when eye pressure is not elevated. In rare families with aggressive NTG, mutations in OPTN or duplications of TBK1 have been found. These mutations also act in a dominant fashion. Because OPTN and TBK1 are involved in cellular stress and death pathways, their discovery showed that neurodegenerative mechanisms, not just high pressure, can drive glaucoma, CYP1B1. This gene, encoding a cytochrome P450 enzyme, is the major cause of primary congenital glaucoma, PCG, glaucoma that appears at birth or in infancy. Mutations in C1b1 are autosomal recessive, meaning a child must inherit 2 bad copies, one from each parent, to develop the disease. Worldwide, CYP1B1 mutations are by far the most common cause of PCG, especially in populations with high family marriage rates. In one large review, over 70 different CY1B1 mutations were identified in PCG patients from many countries. Because this is a well-established cause, any child with true congenital glaucoma is usually offered genetic counseling and testing of CYP1B1, FOXC1 and PAX6. While not classic glaucoma genes per se, mutations in these developmental genes cause anterior segment dysgenesis, underdevelopment of eye angle and iris, often with early glaucoma, e.g. Axenfeld-Rieger syndrome or aniridia. These are autosomal dominant. They remind us that eye development genes can indirectly cause glaucoma. FOXC1 and PAX6 mutations more often present with eye malformations or syndromes, but sometimes the first thing seen is glaucoma. LOXL1, ABCA1, exfoliation glaucoma genes. Another high-profile genetic risk comes from pseudoexfoliation, PXF syndrome, an age-related condition where flaky material clogs the drainage. Variants near the LOXL1 gene were found to confer extremely high risk for exfoliating glaucoma. Interestingly, nearly everyone over age 60 has one of the LOXL1 risk alleles, yet only a minority develop exfoliation syndrome. This cracked puzzle shows that even strong genetic risk may not cause disease unless other factors align. Researchers have also discovered other loci, like ABCA1 and FNDC3B, in genome scans of exfoliation patients, but these confer much smaller risk. In summary, pure single-gene glaucoma is uncommon, perhaps 3 to 5% of adult cases overall. but when it occurs, it often strikes young and can be severe. Knowing these genes helps in rare families or childhood cases. Most glaucoma today is polygenic. Glaucoma as a polygenic disease. For the vast majority of patients, no single glaucoma gene explains the disease. Instead, many common genetic variants each add a bit of risk. Over 120 susceptibility loci have now been linked to glaucoma by large genome-wide association studies. Each locus often contains one or more genes involved in eye structure or nerve health. Here are key themes from these findings. IOP regulation genes. Elevated eye pressure is the biggest modifiable risk factor, so it's no surprise that many risk variants affect pressure control. Variants in or near TMCO1, GAS7, KV1, KV2, ABCA1, AS2, and others have been associated with higher intraocular pressure. For example, Ajibuas found that common SNPs in the GAS7 and TO1 genes were significantly tied to IOP levels, and these same SNPs showed slight associations with glaucoma in combined analyses. TMCO1 and GAS7 are both active in the trabecular meshwork and retina. Variants that determine central corneal thickness, genes like COL5A1 and C1b1, also indirectly affect risk because thinner corneas undermeasure pressure and are a separate risk factor. In short, the genome-wide studies have confirmed that pressure-related biology, water production and drainage, tissue compliance, etc., is driven by many genes. Optic nerve and retinal ganglion cell genes. Other loci influence how robust the optic nerve is or how ganglion cells handle stress. For example, 6 and 8OH7 are developmental genes important for retinal ganglion cell survival, and variants here affect glaucoma risk. SNPs in genes related to the extracellular matrix of the lamina cribrosa, where nerve fibers exit the eye, also show up. Studies have also found variants in genes like CYP1B1, again, and LAMP2 involved in ocular connective tissues, or CKS1B involved in neuron function. The exact functional genes are still being sorted out, but essentially these variants make the optic nerve either more or less vulnerable to damage from pressure or other insults. Vascular and neurodegenerative pathways. Some glaucoma loci overlap with genes known in blood pressure regulation or neurodegeneration. For instance, variants at PDE7B and FMNL2, found in multi-ethnic studies, affect vascular function and have been linked to glaucoma. The THV1, KV2 loci are involved in endothelial function, lining of blood vessels. These findings hint that impaired blood flow, or inner retinal neuron health, is part of glaucoma's complex picture. Other contributing genes. GWAS have also implicated genes affecting eye size and anatomy, which could predispose to angle closure. And metabolism, e.g. cholesterol-handling genes like ABCA1, which may influence nerve health. Often each variant has a small effect size. Only when many risk alleles accumulate do they create a meaningful increase in disease risk. Taken together, the genetic architecture of adult glaucoma is layered. A few rare, large-effect mutations can cause early severe disease, but hundreds of common bumps in the genome each add a whisper of risk. Only in aggregate, sometimes measured by a polygenic risk score, do they become apparent. Genetic risk and ethnicity. Glaucoma's genetic landscape looks quite different in different ethnic groups. Some risk factors are ancestry-specific, European descent populations. Certain glaucoma variants are much more common in people of European ancestry. For example, the MYOC PGLN368 TER mutation mentioned above is essentially seen only in Europeans. Large European cohorts have also found many loci, over 100, through meta-analyses. Because of this, a person of European descent with a strong family history is more likely to carry a known high-risk allele, like in MYOC or KIBCAV2, than someone from some other ancestry. African descent populations. Individuals of West African ancestry have roughly 3 to 4 times the prevalence of POAG compared to Europeans and Asians. The exact genetic reasons are still under study. Some multi-ethnic GWAS show that the overall heritability of glaucoma is high across groups, but many known risk variants identified in Europeans did not fully explain the higher African risk. Studies like the JERA UK Biobank meta-analysis found that African Americans in the cohort had a much higher glaucoma prevalence, 16.1%, than whites, 7.4%. They also showed that within African American ancestry, a greater proportion of African versus European genetic ancestry increased POAG risk. Exactly which genes drive this is a major research focus. It may involve some variants that are common in people of African ancestry, but rare elsewhere. At present, genetic testing panels based largely on European data are less predictive for Black patients. Asian populations, primary angle closure, POAG, East Asians, Chinese, Japanese, etc., have very high rates of angle closure glaucoma, a very different form caused by narrow eye anatomy. Genetic studies in Asians have identified completely different loci associated with angle closure risk. The landmark GBWAS by Vitana et al. Found pleca7, CoL11A1, and a locus near PCMTD1ST18 to be linked to PAC-G in Asians. These genes influence the structure of the anterior segment and iris. Thus, an East Asian person may carry risk alleles that strongly predispose to narrow angles, which are essentially absent in European populations. By contrast, Europeans are more prone to open-angle disease. West Pacific and Southeast Asian groups show similar patterns. Exfoliation, PXF, genetics worldwide. Pseudoexfoliation is very common in Scandinavia, Iceland, and parts of the Mediterranean, and also found in Blacks and Asians, but with different risk alleles. As noted, LOXL1 risk variants are nearly universal across ethnicities. Almost everyone gets one risk copy, but only some develop exfoliation glaucoma. This suggests there are still unknown genetic or environmental second hits. Researchers have noticed that even though the LOXL1 risk allele is common, often 80% in affected people, its penetrance varies. For example, in Nordic populations, the LOXL1 risk haplotype is seen in 80% of PXF patients, but also in 40% of matched controls. In African and Asian populations, the risk and non-risk versions of LOXL1 actually flip, a clear example that LOXL1 alone isn't enough to explain who gets disease. In summary, LOXL1 shows that having a risk allele is almost a given, but disease still needs other factors. Emerging genetic tools and research genetics is moving into the clinic for glaucoma in several ways. Polygenic risk scores, PRS. A PRS combines hundreds or thousands of small risk variants into one score. Recent studies have shown that individuals in the top few percentiles of a glaucoma PRS can have risk comparable to people with a single high-risk mutation. In practice, researchers can genotype a person for known glaucoma-associated SNPs and calculate a risk percentile. APOGs and other groups have demonstrated that a high PRS can identify asymptomatic people who have much higher lifetime risk decades before any damage occurs. For example, one UK biobank analysis found that people in the top 5% of PRS had several times the glaucoma risk of those at median score, similar magnitude to having a positive family history. Each year, more variants are discovered, so PRS accuracy is steadily improving. Very soon, a well-validated PRS, perhaps combined with a patient's age and eye measurements, could be used to flag high-risk individuals for early screening, even before any glaucoma is clinically evident. Pharmacogenomics. Why do some patients respond well to a given treatment while others do not? Studies are beginning to answer this. For example, genetic variants in the prostaglandin F receptor gene, PTGFR, and other drug pathways have been linked to how well a patient's eye pressure will drop on PG analog eye drops. A recent review lists SNPs in genes like ABCB1, a drug transporter, SLCO2A1, GMDS, PTGS1, MRP4, ABCC4, and PTGFR itself that correlate with prostaglandin drop efficacy. In future, we may test a patient's DNA to predict which glaucoma medication will work best or cause fewer side effects, moving toward personalized therapy. Gene therapy and CRISPR. Bench research is actively exploring ways to correct or compensate genetic defects. One strategy is delivering a healthy copy of a gene or protective factor via viral vectors, like AAV, into the eye tissues. For example, animal studies have introduced genes that increased trabecular meshwork outflow or code for neuroprotective growth factors. Another strategy is CRISPR-based gene editing. A dramatic proof of concept came from Jane et al., PNAS 2017, who used CRISPR-Cas9 in a mouse model of MYOC glaucoma. By selectively cutting the mutant MYOC gene in the trabecular meshwork, they relieved ER stress, lowered eye pressure, and stopped further optic nerve damage. This showed it is technically possible to edit glaucoma genes in living eye tissue. Other labs are testing CRISPR on different glaucoma targets and using viral delivery. While human trials are still years away, These advances hint at one day fixing a glaucoma mutation before the patient even has nerve damage. At minimum, these approaches may inspire new drugs that mimic their effects. Genetic testing and family advice. Given all this complexity, what should patients today do about genetics? Here are practical guidelines. When is genetic testing appropriate? Currently, routine genetic testing for adult-onset glaucoma is not standard because most cases are polygenic and current tests are not yet predictive. The rare exception is children or young adults with clear familial glaucoma. Ophthalmologists may order single gene tests or small panels for MYOC, OPTN, CYP1B1, FOXC1, etc. if a patient has very early glaucoma or congenital disease. Identifying a mutation in such cases can inform management and confirm diagnosis. See National Survey Guidelines. For typical older patients with POAG, there's no specific gene test to confirm the disease. The diagnosis is still clinical, eye exam and data. Some specialized genetics clinics may offer broad glaucoma gene panels, but these are mainly used for research or complex cases. Interpreting family history. If you have a close family member with glaucoma, you should tell your eye doctor. You do not need genetic testing. Rather, you should commence routine screening earlier and more often. For example, a child with a parent who has POAG might start seeing an ophthalmologist in their mid-30s instead of waiting until their 50s. Likewise, siblings of glaucoma patients should get checked regularly. Keep in mind that two people can carry the same genetic mutation and have very different outcomes. One person might get late-onset mild glaucoma, while another gets early severe glaucoma. Genetics isn't destiny. Still, knowledge of family history is one of the best risk indicators we have, so err on the side of caution with exams. What to tell your children and siblings? Inform them that glaucoma can run in families, so they need regular eye exams. Glaucoma is sneaky and painless in early stages, so only an ophthalmologist exam can find it before vision is lost. There's no need to alarm, but make sure relatives know to get checked, perhaps with their first dilated exam in young adulthood, if there's a strong family history. Again, even if a parent has glaucoma, it does not guarantee a child will get it. It just raises the odds. They can be reassured that modern treatments, drops, lasers, surgery, can protect vision if glaucoma is caught early. Current limitations. Keep in mind that genetic predictions are still imperfect. A letter or test saying you have a glaucoma gene does not mean immediate blindness awaits. Many carriers never develop disease because of genetic complexity and lifestyle factors. Conversely, a negative test doesn't rule out future glaucoma because polygenic risk and environment play huge roles. Ethical issues include privacy of genetic data and the psychological impact of knowing one's risk for an incurable disease. At present, genetic testing is usually done in research or specialized centers, and results should be interpreted with a counselor or specialist. Patients should not change therapy or stop eye exams solely based on current genetic findings. Looking ahead, routine use of genetics. In the next 5 to 10 years, we expect genetic tools to become more integrated. PRS calculators for glaucoma risk may be validated and offered through eye clinics or even direct-to-consumer genetic services, as is happening in heart disease. Gene therapy for glaucoma is unlikely to be routine in less than a decade, but CRISPR or gene delivery trials may start in the coming years for high-risk cases. For now, the most actionable step is informed screening. Use your family history and eventually your polygenic risk score to guide when and how often to get examined. Conclusion. Bottom line. Genetics matters in glaucoma, but it is not destiny. If you have a family history, you are at higher risk and should get checked regularly. Don't panic if a relative or test shows a mutation. It simply means you and your doctor should be extra vigilant in monitoring eye pressure and optic nerve health. Conversely, even without a known mutation, you can develop glaucoma from the combined effect of many small risk genes and aging. Advancements in gene panels, risk scores, and therapies are on the horizon. Soon, it may become routine to use genetics for personalized glaucoma care, identifying high-risk people before any nerve cells are lost, and tailoring treatments to their genetic makeup. In the meantime, the best strategy remains early detection through regular eye exams, especially for those with any family history of glaucoma. All links to sources are available in the text version of this article. You can find the full article at visualfieldtest.com. Thanks for listening. To check your visual field, click the link at the bottom of this article or visit visualfieldtest.com.